Hello everyone, I am Ikra Sarfraz working as PhD scholar at Department of Zoology, University Faisalabad. I would like to welcome you all in third International Conference on Applied Zoology on the behalf of the Organizing Committee of ICAS 2020. I will be moderator of this session, which will include talks from the fields of biochemistry, genetics, molecular biology, physiology, and microbiology. Let's start this session with our first speaker, Dr. Ahmed Ali from National Agricultural Research Center, Islamabad, and he will be talking about neonatal calf diarrhea, a potent reservoir of multi-drug resistant bacteria and public health hazard in Pakistan. Please, Dr. Ahmed Ali.
Assalamu alaikum everyone. Myself Ahmed Ali. I am serving as a scientific officer in National Microbial Culture Collection of Pakistan located in NARC Islamabad. The topic of my discussion is neonatal calf diarrhea, a potent reservoir of multi-drug resistant bacteria and public health hazard in Pakistan. It is related to one of our ongoing research experiment. Well, calf diarrhea is a very well documented source of zoonosis whereby harmful pathogens can be passed from infected animals to humans and can cause severe illness. And more specifically, neonatal calf diarrhea is a condition where newborn calf animals are susceptible to diarrhea at just their initial period of their lives. And because of this neonatal calf diarrhea, a significant neonatal calf mortality have been reported throughout the world as well as in Pakistan. It is a multifactorial disease and many infectious as well as non-infectious or managemental factors are involved in neonatal calf diarrhea. Presently, dam immunization as well as use of antibiotics are the most exploited prophylactic as well as treatment options to cure calf diarrhea in Pakistan. As most of us very well familiar that antibiotic resistance is a rising public health concern all over the world. And antibiotic resistant bacteria as well as antibiotic resistant genes in their genomes are continuously being reported in many studies of the world. But for neonatal calf diarrhea animals of Pakistan, very limited information is available on the distribution of prevalent infectious bacteria as well as their capacity of resistance to available antibiotics. So the objectives of this study were isolation, characterization, and identification of bacteria from diarrheic animal feces, as well as their antibiotic resistance analysis of those isolated pathogenic bacterial strains. Fecal samples of young diarrheic animals were collected from 6 cities of Pakistan, and a total of 51 bacterial strains were recovered on multiple types of media and identified based on 16S ribosomal RNA gene sequence as well as phylogenetics. And selected strains of bacteria were evaluated for their antibiotic potential or resistance potential. Based on 16S ribosomal RNA gene, all the identified strains belong to 17 genera. And most of the strains, or you can say the prevalent strains that belong to the genus Bacillus, and afterwards Shigella species. And during this study, some of the strains we found were those were candidate novel species of bacteria, and they have a similarity level less than 98%. For example, this one that belongs to Pseudomonas species. And the 16S ribosomal RNA sequences of all these identified strains have been submitted to an international DNA data bank, and accession numbers were also allotted. And very surprisingly, one thing which I want to share with all of you, that based on this sequence 16S ribosomal RNA gene, the pathogenic strains which we isolated from diseased animal feces showed maximum homology with those ones that were reported from human patients. So I mean to say the pathogenic strains which we isolated have a zoonotic potential towards humans, and this is quite alarming. It is basically a phylogenetic tree of one of the candidate novel strain, but it needs further polyphasic taxonomic characterization for its validation. Antibiotic susceptibility testing was performed by this Diffusion method, and you can see that many strains showed resistance to the tested antibiotics. For example, these strains showed against ceftazidime, and many others were. And even some strains showed resistance to multiple antibiotic discs. Similarly, minimum inhibitory concentration analysis was performed against 6 antibiotics and dilutions from different concentrations from 0 microgram per liter to 100 microgram per liter were prepared, and you can see that most of these strains showed maximum resistance to ciprofloxacin. And even one of the strain which belonged to Shigella species showed resistance to a level of up to 75 microgram per liter. And in contrary to this, many of the strains showed sensitivity towards levofloxacin. So we can conclude from these findings that levofloxacin can be used to cure diarrheal infections, particularly bovine diarrhea. Similarly, antibiotic resistant genes presence were confirmed by use of polymerase chain reactions, and for this purpose, reported primers were used, and you can see from this table that many of the strains were resistant or the presence of beta-lactamases genes were in their genome. Similarly, antibiotic resistant genes for mobile cholesterol resistant genes were present, and some were resistant gene or to quinolone resistant genes, and some of them were resistant. You can see that they have resistant genes against multiple antibiotics. So basically these preliminary findings conclude the presence of multi-drug resistant bacteria in fecal ecology of neonatal calf diarrhea diseased animals. And that's why neonatal calf diarrhea disease should be included in the surveillance program of cattle infectious diseases in Pakistan. And antibiotic resistant genes harboring in diarrheic animal feces pose very potential threat to humans, and as these antibiotic resistant genes can transfer from pathogenic to non-pathogenic bacterial species as well by horizontal gene transfer. Similarly, levofloxacin with MIC greater than or equal to 10 microgram per liter could be an effective antibiotic against diarrheic infections of calf neonates. But there should be an alternative source instead of antibiotic usage. Similarly, these studies also furnish presence of candidate novel species of bacteria, which will be validated further by after many other polyphasic taxonomic characterization. So future thrusts include characterization of the fecal bacterial microbiota of healthy as well as direct neonatal calves to get an idea of a true bacterial biodiversity in the whole fecal genome of this ecology. As well as taxonomic characterization of candidate novel bacteria for their validation is also an interest of our program. Use of probiotic bacteria to cure infectious diarrhea in bovine animals. This is the area where we can excel instead of usage of antibiotics. These are some references which I used during preparation of this presentation. And thank you very much for your attention. Thank you very much.
Thank you, Dr. Ahmed Ali, for such an informative talk. Now, I would like to invite the second speaker of this session, Dr. Shehzad Irfan from Government College University, Faisalabad, and he will be talking about peripheral and testicular oxidative stress in varicocele patients may be responsible for primary infertility. Please, Dr. Shehzad Irfan.
Bismillahirrahmanirrahim. My name is Dr. Shehzad Irfan. I'm going to present my oral presentation in the third International Conference on Applied Zoology, which will be held on 7th to 8th December 2020 virtually. The topic or the title of my presentation is Oxidative Stress may be responsible for primary infertility in varicocele patients. First, I will give you a brief introduction. Varicocele is generally defined as a problem in the venous architecture of the testes, and pathologically it is specifically the dilation of the testicular veins in the scrotum, and the outcome of this dilation in the testicular veins usually result in infertility. Statistically, or the prevalence of varicocele is around 35% in men with primary infertility, while the prevalence is raised up to 50 to 80% in men with secondary infertility. The etiology or the cause of varicocele is rooted in the anatomical differences between left and right internal spermatic veins. Absence or inability of the venous walls would result in the increase in venous blood supply, which will cause an increase in the hydrostatic pressure, causing the venous dilatation. This decline in the venous blood flow will slow the testicular circulation, resulting in a decrease in the oxygenated blood supply to the testicular tubules, which ultimately cause hypoxia, and this hypoxia would result in the oxidative stress. This hypoxia, or generally known as stagnant hypoxia, is caused by decreased blood flow and will primarily affect the spermatogenesis, as the aspermatogonia requires a very high level of oxygen for their initial mitotic cell division. In the current study, we aimed to assess the level of oxidative stress at the testicular as well as in the peripheral circulation in varicocele patients which are diagnosed with primary infertility. Over here is a diagram which shows the anatomy of the testicular venous supply, and the varicocele or the varicose veins are usually larger in size because they cannot pump the blood back to the heart. Now, for material methods, after the ethical approval from different ethical committees and the Board of Advanced Studies of the Government College University Faisalabad, and from the Institutional Review Board of Fatima Memorial College of Medicine and Dentistry, Lahore, we included 50 varicocele patients with a mean age of 32. And along with these 50 patients, we also included 10 healthy subjects which were fertile. Now, these 50 varicocele patients were clinically diagnosed as infertile. So, from these subjects, the blood samples were taken, the serum samples were taken, and the blood plasma was analyzed for testosterone, luteinizing hormone, follicle stimulating hormone, uh, cortisol, and for total antioxidant capacity and for the total oxidative status by ELISA. Semen samples were clinically assessed for semen volume, sperm count and concentration, sperm motility and morphology, and tag and toss for the seminal plasma. Now, I will move on towards the results. So, for the endocrine picture, we saw a increase in the cortisol in the patients, while the FSH and LH levels were not very much different. The C-reactive protein were also lower, so there was no chance, or so there was no indication of any infection. The plasma testosterone levels were significantly low in these patients as compared to the healthy controls. The sperm morphology was also significantly decreased. The sperm motility percentage was also decreased. The progressive motility of the sperm in the varicocele patients were also significantly decreased as compared to the healthy controls. The sperm concentration was also found to be extremely low. Total sperm count from each ejaculate was also found to be extremely low as compared to the healthy control, and this is the main reason for the diagnosis of primary infertility. The plasma level of total antioxidant capacity were also found to be low, and the plasma concentration or the plasma levels of total oxidant status was found to be extremely high as compared to the healthy controls. The total antioxidant capacity in the semen was also found to be extremely low, significantly low as compared to the healthy controls, and total oxidative status was also found to be extremely high in the varicocele patients as compared to the healthy controls. So uh, with these results, uh, we believe that the hypoxia is seems to be the main cause of this oxidative damage, as the the testicular tissue or the seminiferous tubules require a very high level of oxygen concentration, which is required for the spermatogenesis. The endocrine profile, if we look at the LH and FSH levels, they do not differ that much. But the levels of testosterone were extremely low in varicocele patients, whereas the level of cortisols were extremely high in these varicocele patients. Now, the precursor for cortisol and testosterone is the same, that is the cholesterol. And it is likely that the Leydig cell, the steroidogenesis which is happening in the Leydig cells, has been affected by the oxidative stress. So the extra amount of cholesterol in the blood has been consumed by the adrenal gland to synthesize the cortisol, which is also known as the stress hormone. Um, for the future outlook, there is one other important point, and that is with the testicular temperature. This hampered blood supply towards the testes also cause an increase in the testicular temperature. Now, for every degree of temperature rise in the testes, the sperm count decreases by 40%. And this low sperm count is the main cause of the male infertility. Um, it is likely that the heat shock proteins, which are generally present in almost every somatic cell, these heat shock proteins, they get activated with a abnormally high or low temperature. So these heat shock proteins, HSP 1790, they might play a role during the process of spermatogenesis in the varicocele patients. Lastly, but not lastly, our results supports this idea that the antioxidant therapy might be helpful in these varicocele patients to recover their lost fertility. Thank you very much, and I think I have exceeded my time limit. Thank you.
Thank you, Dr. Shehzad Irfan from Government College University, Faisalabad, for your nice presentation. And now it's done for our third speaker, Muhammad Asim from University of Agriculture, Faisalabad. And he will be presenting his research work on cryopreservation and quality assessment of semen in Kajli, Lohi, and Thali shape. Please, Muhammad Asim, you have.
Assalamu alaikum everyone. My name is Muhammad Asim and I am PhD scholar in the Institute of Animal and Dairy Sciences in University of Agriculture, Pakistan. My presentation is on cryopreservation and quality assessment of semen in Kajli, Lohi and Thali shape. Actually, I am presenting here my MPhil research. The experiment was conducted on SPU Hyderabad, which is in the district Sahiwal. There were 3 sheep breeds, Kadri, Lohi, and Thali, and 3 animals from each breed were selected for semen collection. Semen was collected with the help of artificial vagina, and 6 to 10 ejaculates from each ram were collected in 10 to 12 days. Semen was evaluated on the basis of macroscopic evaluation as well as microscopic evaluation. In macroscopic evaluation, the color, volume, and consistency of the ejaculates were assessed, and in the microscopic evaluation, the mass activity, individual motility, spermatozoa concentration, motility after dilution, and post-therm motility of the spermatozoa was assessed. For semen cryopreservation, citric acid egg yolk extender was used for the extension of the ejaculates, and the packing of the semen was done in 0.5 ml plain plastic straws. After packing at 4 degrees centigrade, the semen straws were put for 2 hours to bring at equilibrium state. Straws were kept in liquid nitrogen vapors for 7 minutes, and the final cryopreservation was done in liquid nitrogen at minus 196 degrees Celsius. The results show the semen volume the maximum semen volume was assessed in Katli sheep, which was 1.5 ml per ejaculate, and it was significant different from Lohi and Thali sheep. And in the remaining two sheets, it was around 1 ml. The mass activity overall result were very good. It was assessed on the basis of 0 to 5 score, and it was around 3 among all the animals. The maximum mass activity was assessed in Lohi sheep, and the minimum was assessed in the Kajli sheep. If we talk about individual mortality, it was excellent in all the ejaculates from all the rams, and the overall mean was about 80%, and in all the breeds there was non-significant difference measured. The concentration of spermatozoa in million per ml is represented here. Lohi sheep show the maximum spermatozoa concentration in each ejaculate, and it was significantly different from the other two breeds from Kadli and Thali. The spermatozoa mortality after dilution showed excellent result. It was near 75% on the average in all the animals. The post-mortality results were also excellent. It was near 51%. The maximum post-mortality was observed in Kajli sheep, which was 53.13%, while the minimum was assessed 49.38% in the Thali sheep. This table shows the correlation among different semen quality parameters. You can see that the positive correlation between individual mortality and mass activity, mortality after dilution and individual mortality, post-term mortality and mortality after dilution. And a negative correlation was assessed in semen volume and ejaculate number, spermatozoa concentration and ejaculate number, sperm concentration and semen volume, semen concentration and mass activity, and semen concentration with individual mortality. And also a negative correlation was calculated among post-term mortality and spermatozoa concentration. Conclusion. Ram semen can be cryopreserved after extension in standard containing 20% egg yolk and 7% glycerol, and could be successfully used for artificial insemination in sheep. Thank you.
Thank you, Muhammad Asim, for sharing your research work with us. And now we will move towards fourth speaker of this session, Sana Rahim from Kohat University of Science and Technology, and she will be presenting her research work on comparative analysis of in vitro antibiotic potential of selected medicinal plants against E. coli of poultry chicken. Please, Sana Rahim.
Bismillahirrahmanirrahim. First of all, I would like to thank the organizing committee of 3rd International Conference on Applied Zoology 2020 for giving me such a great opportunity to present my research work with the scientific community. I am Sana Rahim, working as MPhil scholar at Department of Zoology, Cohort University of Science and Technology, Cohort. I am here to present my research work, which is entitled as Comparative Analysis of In Vitro Antibiotic Potential of Selected Medicinal Plants against the Escherichia coli of poultry chicken. Starting with the introduction, globally, the poultry industry has experienced massive growth in response to the consumer demand for poultry production over the past few decades. In Pakistan, poultry production is one of the most dynamic and well-organized sector, contributing to 26.8% of the total meat production, 5.76% of the agricultural sector, and 1.26% to the overall gross domestic production. Despite of the scientific development, poultry industry is still in grip of various bacterial, viral, fungal, and parasitic diseases. Among the bacterial diseases, colibacillosis is one of the most frequently encountered problems. It is an acute septicemic disease found in the birds. Colibacillosis is caused by avian pathogenic Escherichia coli, which is a gram-negative facultative anaerobe that belongs to the family Enterobacteriaceae. This bacteria is linked with opportunistic infections in humans, animals, and birds and acts as a secondary pathogen. Avian colibacillosis is considered as one of the principal causes of morbidity and mortality, which is associated with heavy economic losses to the poultry industry worldwide. The use of antibiotics in chickens and the elevated prevalence of bacterial infection is leading to the drug resistance of this bacteria to several drugs. Therefore, utilizing extracts of medicinal plants that might act against these microorganisms are being used by the researchers that is traditionally being given in the poultry feed by the poultry farmers to the chicken also. So, the objectives of my research work were isolation and biochemical identification of E. coli from respiratory system of the infected poultry chicken and to investigate the in vitro antibiotic potential of medicinal plants Kalanchoe agrimontiana, Allobarbadensis, Curcuma longa, Calotropis procera, and the Ilium sativum against E. coli of poultry chicken. To carry out the experiment, the following methodology was followed. First of all, the swab samples were collected from the naturally infected broiler chickens in different poultry farms of the district cohort and then brought to the microbiology laboratory CUST. For the isolation of bacteria, these samples were then diluted in nutrient broth in sterile test tubes and incubated overnight at 37 degrees centigrade. These overnight incubated bacterial broth were then primarily streaked on the McConkey agar media plates and incubated for 24 hours at 37 degrees centigrade. From these McConkey agar media plates, the E. coli suspected colonies were then picked and subcultured on the AFC methylene blue media to obtain a pure E. coli colony. After obtaining the growth on EMB, different biochemical tests were carried out for the identification of E. coli, and all of the results of all of the biochemical tests confirmed the bacteria E. coli. These tests included the colony morphology, gram staining, and biochemical tests such as the triple sugar iron test, citrate test, catalase test, motility indole urease test. The leaves of the Kalanchoe agrimontiana, Calotropis procera, and Allobarbadensis were collected from their natural habitats, while the tuber of the Curcuma longa and bulbs of Ilium sativum were purchased from the local market. For the preparation of extracts, the parts of the plants were shade dried for several days and then grinded, and 20-25 gram of each of the grinded material was then soaked in 1 liter of distilled water. All of the mixtures were then placed on the shaker for 5 to 7 days to for the complete mixing of the extracts. Then the extract was filtered and left at room temperature for several days to evaporate the solvent. The extracts were then diluted further to obtain the final concentrations of 0.8 gram per ml of each of the test extract. The in vitro activity was carried out by the agar well diffusion method. In the in vitro activity, the bacterial broth culture was streaked on the sterile Mueller-Hinton media plates with the help of a cotton swab to form a bacterial lawn. After 30 minutes, equidistant wells were made in the plates with the help of a sterilized cork borer. In this experiment, azithromycin antibiotic was used as a positive control. In the wells, 200 microliter of each of the test extract was filled. The plates were primarily left at room temperature for an hour to allow the complete diffusion of the extracts into the media. And finally, the plates were placed in the incubator for 24 hours at 37 degrees centigrade. After incubation, the resulting diameters of the zones of inhibitions were measured with the help of a ruler and reported in millimeter. Coming towards the results, the following table and graph are presenting zones of inhibition and figure representing the in vitro activity of the medicinal plants. From the table, we can see that the azithromycin antibiotic that was used as control showed a clear zone of inhibition of 27 mm. It was also revealed that the Ilium sativum showed a remarkable antibiotic activity with zone of inhibition of 30 mm, which was quite comparable with the control azithromycin. Allobarbitansis also showed a great antibiotic activity with zone of inhibition of 25 mm. The Calotropis procera and Calantoidegremontiana also showed equal zones of inhibitions of 15 mm. In this study, the Curcuma longa showed no antibiotic activity at all, and the result of Curcuma longa was 0. From the current investigation, it has been concluded that the Ilium sativum, Allobarbadensis, Calotropis procera, and Calantoidegremontiana exhibit antibacterial potential against E. coli of poultry, and it has also been revealed that both Ilium sativum and Allobarbadensis exhibit great antibiotic potential that is quite comparable to the modern antibiotic azithromycin. It has been recommended from my research work that to avoid the drug resistance, the use of antibiotics should be reduced, and medicated feed should be prepared for poultry to cope with the bacterial diseases. These are the references cited in my presentation. At the end of my presentation, I would like to thank all of you for your kind attention and time. Thank you all.
Thank you, Sana Rahim, for presenting your research work at ICAS 2020. And our next speaker will be Dr. Muhammad Mushahid from University of Agriculture, Faisalabad, and he will be presenting his research work on importance of heat shock proteins in livestock vector. Please, Dr. Muhammad Mushahid.
Bismillahirrahmanirrahim. Assalamu alaikum, everyone. First of all, I want to thank the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, for the collaboration for the third International Conference on Applied Zoology and giving us the opportunity for the presentation. My name is Muhammad Mushahid. I am doing PhD in animal breeding and genetics from University of Agriculture, Faisalabad, and I have done my MSc honors from this university, and my topic of research was molecular characterization of heat shock gene HSP70 in nearly rabi buffaloes of Pakistan. I'm here to present about the importance of the heat shock proteins in livestock sector. So what are the heat shock proteins? Family of proteins that are produced under stressful conditions. First, they were reported by the Varicocele and Italian geneticist in 1962. He observed the puffing pattern in chromosomes of Drosophila when exposed to heat. Later, they were also reported in relation to the heat shock, but they were also reported to related with the cold shock. Many members of this family known as molecular chaperones because of stabilizing, correcting, and refolding damaged proteins. During stress, the level of heat shock protein increases in these stressed cells, and it could remain elevated for a long period of time. Several HSPs have been recognized in the absence of any stress, which play important role in cell normal growth and differentiation. So these proteins are produced during heat stress, cold stress, and even during normal functions of the body. Nomenclature. These proteins belong to a multi-gene family and have a wide range of molecular size from 8 to 150 kilodalton. This family has different members which are named according to their molecular weight. For example, HSP 60, 70, 90, and ubiquitin have molecular weight of 60, 70, 90, and 8 kilodaltons respectively. Out of which HSP 60, 70, and HSP 90 are most widely studied HSPs. Important HSPs and their functions. Ubiquitin, target and removal of protein denatured by stress. HSP10 act as a cofactor for HSP60. Low molecular weight HSPs. They help in cytoskeleton, migration, and vascular tone. HSP56 stabilizes the steroid hormone receptor complex. HSP60 molecular chaperones. HSP72 and 3 are highly stress inducible. They are produced during the heat stress. HSP90 important in the steroid receptor complex. So heat stress. The thermoneutral zone is when animal does not have to expend energy to maintain its normal body temperature. In farm animals, 4 to 25 centigrade is considered as TNC. Temperature exceeding 25 will result in heat shock. Animals undergo heat stress when temperature humidity index exceeds 72 and in severe heat shock when rises above the 88. During stressed condition in the cell, HSP interact with the denatured proteins and inhibit the formation of cytotoxic proteins aggregates, thereby maintaining the protein homeostasis of the cell. Heat stress. Heat stress is one of the major issues encountered by the livestock sector, particularly for dairy animals. Its negative impacts and adverse effects are well documented in dairy animals. Reduction in the feed intake, quality of milk, milk production, and fertility. Increased respiratory rates, heart rates, peripheral blood flow, panting activity, and sweating activity are documented. The losses to the US dairy industry were estimated at US 900 million in a year due to heat stress. That's big loss. With increasing global warming, such concerns have risen even more in recent years, and dairy industries are struggling to maximize milk production. Important heat shock proteins during heat stress. Thermotolerance involvement is mainly correlated with HSP70 and 90 in livestock species. HSP70, namely HSP71, HSP72 is reported to be the most abundant and temperature sensitive. In farm animals, elevation in HSP70 and 90 was observed in sheep, buffalo, cattle, broiler, and goats. Here it is the cellular and molecular mechanism of the production of HSPs. When there is a heat stress, the cellular and molecular response triggered. What happens? Cellular damage and protein denaturation starts, and misfolding and migration of misaggregation of the proteins occurs. So what happens here is heat shock response also triggered during this condition, and it includes heat shock element, heat shock factor, heat shock proteins. And what heat shock proteins do, they prevent the misfolding and misaggregations of enzymes and proteins and help in the cellular adaptation and homeostasis of the cell. Selection of the thermotolerant animals. The gene that is related to thermotolerance is the heat shock gene. HSP70 and 90 are most abundant. These can be used as a marker for the marker-assisted selection and genome-wide selection. Development of SNPs using these genes will be helpful for the selection of thermotolerant animals. My conclusion is heat shock proteins are important during the stress and even normal cell functions. HSPs play an important role during heat stress in livestock animals. In livestock, HSP70 and HSP90 are most abundant during the heat stress. HSP70 HSP genes can be used as a marker for the selection of thermotolerant animals. Thank you for your attention.
Thank you, Dr. Muhammad Mushahid, for such an interesting talk. And now it's turn for Dr. Ghulam Hussain from Government College University, Faisalabad, and he will be talking about evaluation of functions, retrieval, ameliorating effects of indigenous flora following an induced insult to a peripheral nerve. Please, Dr. Ghulam Hussain.
Assalamu alaikum. Good morning. I am Dr. Ghulam Hussain from Department of Physiology, Government College University, Faisalabad. Before starting my presentation, I would like to pay thanks to the organizer of the Third International Conference on Applied Zoology 2020. The topic of my presentation is evaluation of function retrieval ameliorating effect of indigenous flora following an induced insult to a peripheral nerve. Let's have a glance on the background of the work. As it is known that the command and control system of our body is the central nervous system, and it sends instruction in the form of electric impulse to the organs by using a complex network of the wires that are known as the peripheral nerve. Any injury as a result of sharp electrocution, road accidents, gunshot, and any other trauma can result in an interrupted electric impulse transmission. And that may cause the loss of instruction to the organ. And in the absence of this instruction, the organ that are usually muscles, they become functionless, and this loss of functionality can be partial or can be complete depending upon the severity of the injury. Although after an injury, the peripheral nerve can regenerate, but the process of regeneration is very complex that follow a complex cascade of the events, starting from the injury to the retrieval of the function. And if we summarize these events in the form of the figure, we can come to know that the loss of conduction starts after an injury, and after this process, the Valerian degeneration takes place. That is the degeneration of the distal end of the axon, and after this process the axonal regeneration takes place. And if the regeneration is complete, then the functions of the organ can be regained, but unfortunately this process is not very simple, and the regeneration is very, very slow. That may take weeks and even months and even years. So how we can overcome this problem to gain the functional retrieval complete and perfect? The available treatment at the moment, they are symptomatic treatment. There can be surgical approaches, and there may be supportive therapies and some other approaches, for example gene therapy and stem cell therapy, etc. But these processes do not provide the complete and the perfect functional regain due to various factors. For example, they are time consuming, expensive, and in case of surgery, the unavailability of the donor is also a big problem. Then what's the reason how we can accelerate this rate of regeneration? Here come the entry of the natural products. As we know that the plant have been a rich source of new compound that can be used as a health promoting factor since the beginning of the history. So we can speculate that these compounds from the plants can be a miraculous candidate that can accelerate this deadly slow nerve regeneration process. To achieve this objective, our labs follow a screening process accompanying a series of the experiment. By employing these approaches, we try to evaluate plants that can be most effective. And here is a set of experiment that we apply starting from the evaluation of the plant, and that can lead to the identification of the potent bioactive component of the plants, and that will be our interesting target as a therapeutic intervention in the future. Based on these observations, this work, we have identified the most important plant as the Moringa oleifera. This plant is also known as the tree of life, and there is a huge data on this plant. And one important factor is that this is the most important, most nutrient-rich plant yet discovered, based on the various other factors. For example, the presence of essential amino acids, several minerals, and it's also a rich source of different vitamins. And here is a summary of its important feature. For example, it contains 25 times more iron, 70 times calcium, and so on, so on. Based on these factors, this plant has been studied extensively, and here is a summary of all these beneficial effect of this plant. Some of the key feature can be antidepressant activity of this plant, antioxidant, and increased mental clarity. Due to its relevance with the central nervous system activity, one can raise the question that the can moringa oleifera accelerate the motor function recovery after a mechanical insert to the nerve. To address these questions, we follow a well-established model of sciatic nerve injury in our lab, and by which we induce the sciatic nerve by using a forceps method. And this model mimic the actual symptom as we observe in human being. Okay, by employing this model, we perform a project timeline. We induce sciatic nerve at this point at time point 0, and after this induction of the sciatic nerve injury, we also start offering the plants that was the moringa oleifera in our case. And after this time point, we try to evaluate the functional retrieval by using different parameters. These parameters can include the behavior analysis, biochemical analysis, histopathology, and messenger RNA level at all. For the time being, we have evaluated this plant on the basis of the behavioral parameter. These can be food intake, weight analysis, walking track analysis, grip strength, chemoception, and thermoception. Based on these experiments, these observations, we have obtained some finding, and they are: the number one is the food intake, and we could identify that the food intake was normal in both groups, in the presence of the moringa oleifera that was mixed in the diet, and there was no effect of this moringa oleifera on the weight change in both groups. And when we measured the grip strength, we could observe that in the treated group, the rate of functional retrieval in the form of the muscle grip strength was ameliorated in the treated group. And the similar finding were observed in the walking pattern of these animals. And we could note that the treated animal exhibited an accelerated functional retrieval, and the response of these animals to the thermal stimulus has also improved, and the latency period decreased in the treated group as compared to the control group. And at the case of the muscle mass, the treated animal also exhibited an improved muscle mass ratio. At the systemic indexes, we could note that there was improved antioxidant capacity of the treated animal. Based on these observations, we could conclude that the moringa oleifera offers emulating effects on the functional retrieval for a mechanical insert. Although this is the preliminary information, and there are some important things that should be done to verify this finding. And that can be: what is the happening at the gene expression level, what is the response of these muscles fiber to the treatment, and then what are the actual constituent of this plant, and these molecules should be characterized. And then what are the signaling pathway that can play a role in this response. So we speculate from this observation that this plant should be evaluated for the further effects. Thank you very much for your kind attention.
Thank you, Dr. Ghulam Hussain, for such an informative talk. And now I will move towards seventh speaker of this session, Dr. Asma Ashraf from Government College University, Faisalabad, and she will present her research work on evaluation of antioxidant and anti-diabetic potentials of Tabernae montana, Divaricata, and therapeutic application. Please, Dr. Asma Ashraf.
In the name of Allah, the most beneficent and the most merciful. The topic of presentation is evaluation of antioxidant and anti-diabetic potential of Tabernae montana, Divaricata, and its therapeutic applications. As we know that diabetes mellitus is a disorder with a complex and multi-phase etiology. Type 2 diabetic mellitus caused by the resistance of insulin with increased pancreatic insulin. Therefore, insulin fails to maintain the body metabolism. According to International Diabetes Federation, it is estimated that worldwide 1 in 11 adults with the age of 20 to 79 years old has diabetes mellitus. However, it is experienced by 80% in low to middle income countries as diabetes account over 3.8 million deaths annually. That's why it is the fifth leading cause of mortality. Oxidative stress plays a major role in diabetic physiopathology. The mitochondrial system strongly links hyperglycemia to increase the development of superoxide. The elevation of reactive oxidative species level in diabetes may be due to reduced destruction and or increased production of antioxidant enzymes. Medicinal plants are important source of pharmacologically active antioxidant. Different metabolites of plants like phenolic having antioxidant properties show a crucial role in alpha amylase and alpha glucosidase inhibition. Therefore, they have the potential to contribute the management of type 2, type 2 diabetes mellitus. In this research, we have used the plant, which is Taberni montana divaricata, which is belong to the family Apocynaceae. This plant is commonly known as Chandni or crepe jasmine. This plant is widely used as a herbal medicine in tropical areas. In India, the application of this plant may be classified as a panacea for gastrointestinal infection, urogenital and skin infections. The root, leaves, and flowers of this plant are used in the treatment of skin and scorpion poisoning. The objective of the current study is to evaluate the secondary metabolites and in vitro biological properties like DPPH free radical scavenging, enzymatic antioxidant properties, hemolysis, anti-diabetic potential of methanolic extract of T. divaricata flowers. This is the layout of methodology followed by us. First of all, we have collected the flowers of plant. We have dried the flowers and crushed them into a fine powder. The extract was made by using the methanol as a solvent. In phytochemical analysis, we have determined the two major components, the total phenolic components and total phenobolide contents. In antioxidant assay, we have done DPPH free radical scavenging assay and some enzymatic antioxidant activities like catalase, peroxidase, superoxide dismutase. To check the cytotoxicity of the plant, we have following the procedure of hemolytic activity. To check the anti-diabetic potential of the methanolic extract of the plant, we have followed the three inhibitory assay, which are alpha amylase inhibition assay, intestinal alpha glucosidase inhibitory assay, and glucose uptake in the yeast cell. The result of the current study show that the plant have high content of total phenolic contents, which are 52.32 microgram, and the range from and the total phenolic content showed by the these are the result for biological activity. The plant extract show 15.9% DPPH free radical scavenging activity in comparison with the standard ascorbic acid. These are the result for some enzymatic activity. The result from antioxidant activity show that the free radical scavenging along with the enhanced in physiological antioxidant enzyme study show that the plant has capability of inhibition of both alpha amylase and alpha glucosidase enzyme. As we show that at the concentration of 50 microgram per ml, the plant show the activity of inhibitory activity of 14.32%, and as the concentration of the plant raised, the activity shown by the inhibitory activity shown by the plant is also raised. At the same concentration, the inhibition of the inhibitory activity of the plant also raised, and alpha in this activity the plant also show the dose dependent inhibition. In the next assay, the extract was studied for its efficacy to uptake of glucose by yeast cell using many molar glucose concentration. The uptake of glucose was evaluated by incubation of yeast cell in the different molar concentration of glucose like 5, 10, and 25 millimolar, and the plant extract solution while using the reference or standard drug metronidazole. And the result of the study show that By rising the extract concentration and the metronidazole, the uptake of glucose percentage was also increased. This is shown. This is showed also an inverse relation with the glucose molar concentration as described in the figure 5A to figure 5C. The result of this study conclude that the flower of T. divaricata are the potential source of natural antioxidant. The free radical scavenging along with the increase in enzymatic antioxidant enzymes also is due to the action of phytochemical of this plant. This traditional alpha amylase and alpha glucosidase activity compared with the common drugs. The present study results would be useful for future in the development of nutraceuticals and pharmaceutical drugs. Thank you for your attention.
Thank you, Dr. Asma Ashraf, for sharing your research work with us. And now it's time for Dr. Mehvi Salman from Government College University, Faisalabad. And she will present her research work entitled Bisphenolate, a major bioactive metabolite produced by Lactobacillus corniformis BCH4 with antibacterial, antioxidant, and larvicidal activities. Please, Dr. Mehvi Salman.
Bismillahirrahmanirrahim. Present my paper in third international conference on vectors of many diseases. In addition, many synthetic insecticides such as organophosphates are used to control the biological vectors, but their use as an insecticide has been prohibited due to risk issues. Therefore, we are using natural bioactive metabolites exhibiting unusual structures isolated from microorganisms have been playing a significant role in drug discovery. Therefore, we are using lactic acid bacteria and their metabolites because they have potent bioactivities like antimicrobial, antioxidant, and larvicidal activities. We have and the isolated compound that is a bis-2-ethyl xylphthalate has been reported as an potent bioactive secondary metabolite naturally produced by microorganisms. These are the main objectives of the study, that is the fermentation of Lactobacillus coroniformis BCH4 for isolation, purification, and structural elucidation of the respective compound. Potential of BEHP, that is the bis-2-ethyl xylphthalate, to determine antimicrobial, antioxidant, and mosquito larvicidal activity. That is the overall methodology. First, we have performed fermentation of Lactobacillus coroniformis BCH4 that was previously isolated in Department of Biochemistry. Then we perform extraction by using ethyl acetate solvent. Then we do purification by silica gel column chromatography, and the structural elucidation has been determined by GCMS and the respective functional groups by FTIR. These purified fractions then undergo for the determination of biological potential by performing antibacterial, antioxidant, and larvicidal activity. Now we move to our results. That is the culture that we obtained of Lactobacillus coroniformis BCH4. Then we do culturing and harvesting, and the supernatant we got is passed through 0.22 micron millipore filters. Then we got filtration after syringe filtration process. Then we do extraction, and we got this oily upper layer which was transferred to silica gel column, and we got the purified fractions. These were further employed to determine antibacterial activity. We got four fractions, sorry. Then we selected F2 fraction, which is more resistant, shows more resistance against E. coli and Staphylococcus aureus. This fraction was further undergone through GCMS, and we got a single peak, and which was the structure was further determined as bis-2-ethyl xylphthalate. And the functional groups of this compound has also been determined by FTIR spectrum. That is the FTIR spectrum of purified BEHP metabolite. This is the antioxidant activity that was performed in different concentrations in microgram per ml, that is a 25, 50, and 100 microgram per ml. We got maximum antioxidant activity at this 100 microgram per ml concentration. Next, we have also performed mosquito larvicidal activity. That is the mortality mean of Culex quinquefasciatus larvae at different time intervals and concentrations of BEHP. That is the concentration dependent manner, 50, 0 ppm, 50, 100, 150, 200, and 250 ppm concentration with different exposure of time, that is a 24 or 48 or and 72 or. And we got 100% mortality at maximum concentration, that is a 250 ppm after 72 or. 100% mortality of Culex fasciatus larvae. Next, we also checked the toxicity of BEHP against Culex fasciatus larvae, and we observed this toxicity by determining LC50 value after 24, 48, and 72 hours. And the 72 or after 72 or we have a least value of LC50. As the toxicity of the compound increases, the LC50 value of the respective compound decreases. So after doing that, we also calculate the effect of different concentration of percent inhibition of acetylcholinesterase enzyme activity in C. Culex, Culex fasciatus larvae after 72 hours. We only performed this experiment after on the component we got after 72 hours. Therefore, after 72 hours at 250 ppm, we got maximum or 100% mortality. So we got these values, and the maximum acetylcholine esterase inhibition was observed after 250 ppm concentration at 250 ppm. So that is the conclusion. The production of bisphenol phthalate by L-choliniform BCH4 found as potent bioactive metabolite exerting antibacterial antioxidant activity. Moreover, this compound also have potent against larvae of Culex species. It also modulate the ACH enzymatic activity, which causes the death of C. Culex larvae. Our research collaborators, these are the different respective teachers that are counted in our research as collaborator, and we also want to acknowledge the Department of Bioinformatics and Biotechnology and the respective persons, and thank you so much for your attention.
Thank you, Dr. Mehbi Salman, for your interesting talk. And now it's turn for Syed Ataur Rahman Shah from Cohort University of Science and Technology, and he will present his research work on effects of atrazine on blood biochemical and hematological indices of grass carp. Please, Syed Ataur Rahman Shah.
Assalamu alaikum. Hello everyone. Welcome to the third International Conference on Applied Zoology, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. I'm Syed Ataur Rahman Shah, belongs to Department of Zoology, Cohort University of Science and Technology, CAST, Cohort. I completed my work under the supervision of Dr. Farmanullah Davar on the topic effects of atrazine on blood biochemical and hematological indices of grass carp. Introduction. We are familiar that Pakistan is an agricultural country where different herbicides and pesticides are used for the control of herbs and pests. So here, atrazine is also one of the commonly used herbicides used for the control of annual grasses and broad-leaved weeds. Atrazine is not degraded by microbial or hydrolytic process. So because of that, atrazine is present in the soil due to irrigation and water or raining. The atrazine run off into water bodies. When it run off into water bodies, so we know they are aquatic animals, and we also familiar that atrazine is highly toxic and carcinogenic in nature. So it have adversely effect upon the immune response, blood indices, growth, reproduction, behavior, and physiology of the fish. What was the objectives of the current study? So there was two objectives in mind. The first one was to analyze the potential effects of atrazine on biochemical indices of grass carp, and the second was to investigate the potential effects of atrazine on hematological indices of grass carp. material and methodology, I completed my work in the two steps. And the first steps, collection and acclimatization of grass carp. Grass carp was collected from the local fish farm from Peshawar and then transported to the Aquaculture and Fishery Laboratory Department of Zoology, Gujarat. After that, the fishes were acclimatized in the water tanks for 2 weeks, and after that, atrazine dilution, technical grade atrazine 97.5% were used for the stock solution, in which 5 ml of atrazine and 5 ml of 80% acetone was mixed, and after that, from this stock, 1 ml of the solution was taken and it was mixed with 9 ml of distilled water. From that 10 ml solution, different concentration was given to the fish. So we made acute toxicity experiment, and that experiment we made 5 groups. One was the control and the other 4 was the atrazine treated groups, in which different concentration 50, 100, 150, 200 microliter per liter were given to the fish. After that, and the second step, we find first the LC50 or lethal concentration of 50 on the 50% population of the fishes were determined, and also the hematological and biochemical component was also determined. So going towards the results. And the results, I will only share with you 2 components from the hematological and 2 components from the biochemical due to shortage of time. So the first one is RBC, red blood cells. And the graph, you can see that the red blood cell concentration in the control are different from that of the treated groups because, and it significantly decreased. The reason behind it is because atrazine is toxic to the fish and it affects different cells of the fish, behavior, physiology, biochemistry of the cells. So because of that, the hematopoietic tissues also got affected because of the toxicity of atrazine. When they affected, so it not produce more RBCs into the bloodstream. So because of that, the RBCs concentration, the RBCs level decreases, and different treatment groups with different concentration and different times. The second one is the white blood cell WBCs, and here we see in the graph WBC level was increases from control to the different groups significantly increased because when the foreign particle or the foreign toxin enters the body, so the lymphatic system produces lymphoid cells, and these cells fight against these toxins. So here WBC level increases, which means that this WBC's fight against the toxin and its level was increases. The third step is the glucose. Sorry, the third component is the glucose. And we see in the graph that the glucose level was significantly increasing. Why it is increases? Because we are familiar that blood glucose considered as indicator for stress response in fish. Hyperglycemic condition induced by environmental pollution might produce stress response in fish by the inhibition of the gluconeogenesis process, which enhances glycogen breakdown in the liver and consequently elevates blood glucose level. Another reason may be the hyperglycemic state could be explained as result of the increased secretion of catecholamines from the adrenal medulla, which in turn enhances the breakdown of glycogen, and because of that, the blood glucose level will increase. Why it increased? Because of the toxin atrazine given. So it's all about the glucose, and the graph we can see a significant increase was come. The last one is the cholesterol, and from the graph we see that the cholesterol and the control group was increased and it was then significantly decreases. What's the reason? Because we are familiar that cholesterol is the best material for all steroid hormones. When it's increased due to its cortisol synthesis, then a large amount of cholesterol is needed. Therefore, the reduction in the amount of cholesterol may be related to its utilization and the manufacture of cortisol arising from stress created by the toxin atrazine. And because of that, we see that the cholesterol level was decreases because it was used in the cortisol synthesis. Thank you so much for your attention.
Thank you, Sayyid Ataur Rahman Shah, for your presentation. And now it's time to move towards our last speaker of this session, Shumaila Zafar from Government College University, Faisalabad. And her topic of presentation is Calotropis procera bestows ameliorating effect on the mechanically inserted sciatic nerve in the mouse model of peripheral nerve injury. Please, Shumaila Zafar.
Okay, in the name of Allah, the most merciful and beneficent. Assalamu alaikum, and thank you so much, ICAS, for giving me a chance to present with the topic Calotropis procera bestows ameliorating effect on the mechanically inserted sciatic nerve in a mouse model of peripheral nerve injury. I am Shumaila Zafar, PhD scholar from Department of Physiology, Government College University, Faisalabad. As we know that the nervous system being a controller and coordinator of all the bodily functions, any kind of interruption in any parts of can result in the loss of the bodily functions. Particularly if you talk about the peripheral nervous system, which comprise of the long distant bundles of axons and innervating the target tissues or muscles. So any kind of interruption in this in these tracts result in the denervation of the muscles, which, and if it persists for a long time, it leads to the muscular atrophy. Although nerves have capability to regenerate itself, yet this process is too much time taking that to accomplish, that it result in the irreversible irreversible revival of the target organ or muscles. So there are so many causes as a result of which peripheral nerve injuries develop, like road accidents, sudden bomb blasts, sudden falls, lacerations, and which result in the compression and restoration of the nerves. Even in some kind of endogenous diseases like presence of tumors, which exert pressure on the nearby nerve, can lead to its injury and loss of functions. If we talk about, say, therapeutic interventions, and most commonly surgery is the surgery is required, yet it doesn't ensure the 100% recovery even after surgery. Moreover, different symptomatic therapies have been given to heal the relevant issues like inflammation or pain, etc. Yet, if we talk about any agent or any drug that particularly accelerate the functional recovery after nerve injury by speeding up the process of regeneration to re-innervate the distal organ or muscles, then the answer is still no. Although researchers are continually working on this aspect, yet we don't have even a single agent available which targetedly affect or speeding up this nerve regeneration. So plants are already well reported for their role in healing a lot of injuries. So Calotropis in dealing a lot of injuries and diseases. So Calotropis procera is among such plants which have very, very effective constituents and having potential to heal a long list of diseases. Like it has well-reported role as an antimicrobial, antioxidant, anti-malarial agents. It also has been reported to have anti-inflammatory and anti-nociceptive effects. So based on the its well-reported effects in different aspects, if we talk about its role in curing the nerve injuries or in speeding up the nerve regeneration and increasing the rate of recovery after such kind of peripheral nerve injuries, then there is no even a single study available discussing about this aspect of this aspect. So we decided to design a study to find out its role in accelerating the functional recovery after peripheral nerve injury. For this purpose, we use an animal model of in which sciatic nerve injury was induced in the sciatic nerve of one of the hind limb. For this, we took albino mice, adult healthy albino mice, and divided them equally into two groups, and induced this injury. And we performed different behavioral parameters both before and after nerve injury induction. We did food intake and weight analysis to find its role. to find and to compare the effect of incorporating the treatment like this plant in the food and animal behavior and animal behavioral changes due to the addition of this plant in the food. So we performed the walking track analysis and grip strength to figure out the extent of recovery in motor functions. And similarly, we performed pinprick and hot plate test, which are particularly representing the sensory function restoration in both groups. We also performed different, at the end of the study, we take out the serum samples and perform different oxidative stress markers like turbulent external capacity and dose and activity of the aryl estrane and pyrosinase enzymes. And this is the timeline which we follow in the whole study, like for conducting the different parameters both before and after engine induction. Now come towards the results. The results of body mass and food intake clearly shows that the incorporated plant material in the animal's diet did not change the animal behavior or animal eating pattern due to this addition. Because both in body weight analysis, animals grow in the normal way as they grow under ordinary conditions in both groups. Similarly, after their diet intake pattern was also appeared to be non-significant on all time points. If we talk about its motor functional recovery, the analysis of the grip strength clearly shows that the treated group have shown highly significant improvement on day 9 and even on day 9 and day 12 as compared to the control. And similarly, the SFI, which is showing the static functional index, we found very significantly improved value of SFI on day 6 and day 9 in the treated group. Basically, the static functional index is the main indicator showing the animal hind parts moving towards its normal position. Sensory functional restoration as assessed by the paw withdrawal latency from the horse surface and as a result of pricking the skin by pain. So we found very significantly earlier paw withdrawal in animals treated with this plant as compared to control, and similarly the value of TAC in treatment group and reduced value of TOS, that is reduction in the total oxidant status in the treated group. Similarly, we found comparatively increased activity of aryl estrane and pyroxenase in the treated group, which particularly indicates which particularly indicates the antioxidant effect of this plant in the treatment group. Concluding remarks. So from the results, it appears that the calotropins could have a potential to accelerate the functional recovery following PNI, and the results of the oxidatory stress parameters shows that this plant have the oxidative stress combating effect, and it can be assumed that this speedy recovery in the treated group may be due to the controlling the oxidative stress in the animals. So on the basis of these conclusions, it can be prospected that this plant should be completely characterized to identify the potent bioactive component, which were particularly playing the main role behind this potential, and moreover the mechanistic elucidation to using the advanced technologies should be done to get favorably supportive data to consider these components for future drug development and preclinical study. That's all about my work. If you have any questions, you are most welcome. Thank you so much for your attention.
Thank you, Shumaila Zafar, our last speaker of this session. Hope you all have very productive time during this session with us. And now our next session will start after 30 minutes of break at 2 p.m. And Ms. Rabia Zara will be the moderator of our next session, which will include entomology, toxicology, and pathology. Stay tuned with us.